program over to Dr. Felicia Keising, co-director of the Tick Project, Carrie Adjunct, and David and Rosalie Rose, Distinguished Professor of Science, Mathematics, and Computing at Bard College.
Thank you, Lori. Welcome to everyone near and far. There is quite a far-flung crowd tonight, judging from what's in the chat. Thank you for joining us this evening. As Lori said, I'm Felicia Keising. I'm a professor of biology at Bard College and one of the two directors of the Tick Project. I'll be moderating the event this evening, and I'm joined by my colleague, Rick Ostfeld, the other director of the Tick Project. Rick will be giving a short presentation about the project, which will last about half an hour, and we'll reserve the rest of the hour for questions from you, the audience. You can ask your questions using the Q&A feature at the bottom, or in some cases, the top of your screen, as Lori said. And so without further ado, let me formally introduce Rick Ostfeld, Senior Scientist at the Carey Institute of Ecosystem Studies and co-director of the Tick Project. Rick.
Thank you, Felicia, and thank you, Lori, for starting things off. And thank you all for joining us on this webinar this evening. It's much appreciated that you took the time out of your busy schedules to come and listen to us describe the rationale, the design, and the results, the major results of the Tick Project. Earlier this afternoon, I counted up the cumulative number of years that Felicia Keising and I have been engaged in research into the ecology of tick-borne disease. And I was shocked by the number, which was 52. So that's a cumulative number of years. It doesn't consider the overlap between us. But it's a long time to be working on this problem, and the problem deserves it. It requires a lot of time and a lot of effort. Most of the effort that we have engaged in over that cumulative 52 years has been what would generally be considered basic research. We're trying to understand how the tick-borne disease systems operate. Who are the important species? How do they interact? What gives rise to variation from one place to the next in the number of infected ticks that are crawling around and can potentially make us sick? What gives rise to some years being much riskier than other years? How does that change through time? So that's our research has been directed at trying to generate a basic understanding of how nature works, this particular corner of nature, which is the ecology of tick-borne disease. About 6 years ago, we were able to take advantage of an opportunity to engage in probably what is the most applied research that either Felicia or I have undertaken in our careers. And by applied, what I mean is that we were trying very hard to take advantage of the knowledge that had been generated during this long, intensive phase of basic information gathering about how the system works, and attempt to use that, apply it to solve problems. And the problem we were attempting to solve, or at least contribute strongly to solving, is that of tick-borne disease, particularly Lyme disease. We're trying to reduce the incidence and the risk of Lyme disease in our communities. And that is what the Tick Project was designed to do. So without any further ado, I'm going to share my screen. And start with the slides. And I'm assuming that this can all be seen. Move some distracting images like of myself out of the way. So with the Tick Project, we designed this study to test environmental interventions to prevent Lyme disease and other tick-borne diseases in our communities. This is an increasing problem in the United States, in other parts of North America, in Europe, in parts of Asia, in North Africa. So over a very wide distribution, we've seen the emergence and very potent increase in the number of cases of tick-borne diseases. And here in the northeastern US, southeastern Canada, the upper Midwest, and elsewhere in this country, we have sort of the big three of tick-borne diseases, where Lyme disease is the most prevalent of all, but it's not the only one. We also have anaplasmosis and babesiosis, and these are increasingly important problems. There are increasingly higher and higher incidence rates in our communities across the country. And I'm showing data from governmental agencies on the number of cases of reported cases of Lyme disease, anaplasmosis, and babesiosis. The exact numbers are not that important for reasons I'll describe in a moment, but the trends are what I want you to understand. We have increasing trends of all three of these diseases that are very alarming. They're getting worse and worse all the time. In the case of all of these diseases, the actual counts of human beings who come down with one of these diseases every year are known now to be gross underestimates of the real burden of these diseases. And in the case of Lyme disease, that's been estimated fairly rigorously at about a factor of 10. So we are only recording about 10% of the actual number of cases of Lyme disease when we see the CDC statistics on their website, for instance, which is where these data come from. The current estimates are that there are between 400,000 and 500,000 new cases of Lyme disease in the United States every year. And that burden of Lyme disease is inexorably rising, as is the case with anaplasmosis and babesiosis as well. And they are all transmitted by a bite of the same tick, the black-legged tick, shown a little bigger than life size in the bottom of the slide. One of the main reasons why Lyme disease and other tick-borne diseases have been increasing in their public health burden is because they are expanding in their geographic ranges. This slide shows the situation at the turn of the 21st century, where Lyme disease occurred in a band in the Northeast along the Atlantic seaboard, and in an area in the upper Midwest, spanning the border between Wisconsin and Minnesota. A mere 20 or so years later, not even 20 years later, this is what the distribution of Lyme disease looked like, where it had expanded and subsumed all of New England, all of New York, all of Pennsylvania, gone down into Maryland and Virginia, into West Virginia, and similarly had expanded dramatically in the upper Midwest. So I'm just going to go back a second so you can see the 2001 distribution. And then only 16 years later, it had expanded into dramatically larger areas, and that is continuing up to the present. So Lyme disease has been known to be a very important and difficult problem, public health problem. And as is the case with many of these infectious diseases, there are sort of three main arms in our armament to try to protect the public against these diseases. We have prevention, diagnosis, and treatment. Prevention generally includes vaccination, which in the case of Lyme disease was available for a few years back in about 1999, 2000, 2001, but it has not been available for us since that time. Prevention largely has been explored in the sense of trying to reduce tick populations so we reduce our actual exposure to the pathogens that are transmitted by these ticks. Then we also have diagnosis. We are using 50-year-old technology still to try to diagnose Lyme disease. So there is a considerable amount of effort that is being used to try to improve our ability to determine who's sick with Lyme disease, who needs to be treated. If we know more accurately who is ill with Lyme or another tick-borne disease, then we need to also do a much better job in treating those people to make them healthier. And again, we're using several decade-old technology to treat Lyme disease. That too is the subject of a great deal of very energetic research. So I would argue that in the past 5 to 10 years, there's been actually a largely stepped up effort to understand how to better diagnose and how to treat tick-borne disease. But that approach is almost accepting that we're going to get exposed rampantly to tick-borne diseases, and all we need to do is to be able to figure out who's sick and figure out how to treat them. And that, to Felicia and me, was unacceptable. We were concerned, that there was so little emphasis on prevention in the form of controlling tick populations so that we could avoid the need for so much diagnosis and so much treatment in our afflicted population. So what I've argued so far is that we have a huge problem. We have a rapid increase in the incidence of tick-borne diseases and in their geographic range. The estimated cost just in healthcare costs alone of Lyme disease is about $1 billion a year in the United States, and that's not accounting for our rampant inflation that we're experiencing right now. And it doesn't account for other economic costs such as lost productivity, lost recreational opportunities, and even declining property values as a consequence of high risk of tick-borne disease. As I mentioned, there are no vaccines available, and diagnosis and treatment are emphasized, but they remain problematic. Importantly, effective tick management is very poorly supported. It's not very well understood how to do this, and the history has not been very promising. So that set the stage for the Tick Project. It's a little surprising that we haven't been more effective at understanding ways of killing ticks in order to protect our public health, because it turns out it's not that hard to kill ticks in the environment. There are several methods that have been developed that are known to be able to do so. Largely, there are chemical sprays. These are the best known, the best studied. So these are typically synthetic pyrethroids or organophosphates that are sprayed in the environment, either using handheld sprayers or high-pressure truck-mounted sprayers. They're quite effective at killing ticks, but unfortunately, they are also very potent in killing other organisms that we don't necessarily want to kill. So these would be non-target effects. They're devastatingly effective against honeybees and other kinds of pollinators, against butterflies, against predatory insects that help control pest insects, and they can be highly toxic or at least moderately toxic to vertebrate animals, including us. There are also biocontrol sprays. So these are not chemical sprays, but they are actually solutions in which microscopic organisms, microbes that make their living by attacking ticks are present in the sprays. They tend to be more specific, at least some of them are more specific against ticks. They have fewer of these non-target effects. They generally are quite non-toxic to our children, our pets, to us, to other vertebrate animals. They tend, though, to be somewhat less potent than the chemical sprays are. And then lastly, there are host-targeted methods of killing ticks. These are generally chemical, like the chemical sprays that I mentioned, typically different classes of chemicals, but they are not broadcast into the environment. They're not sprayed. Instead, they are used in devices that attract certain species of hosts, like small mammals or like deer, and deliver the chemical only to those hosts. So we don't see those non-target effects and the potential toxicity. When we use these methods of killing ticks, there are numerous sort of small-scale pilot studies that are used to assess them. Tick numbers are generally reduced by at least 50% and sometimes more like 90%. So they tend to be fairly effective in these pilot studies. Unfortunately, that information comes from studies that are typically done at very small scales with low levels of replication. So our ability to infer whether they can really be effective in protecting public health is generally quite limited. Many of them have design flaws that include the poor use of controls and the potential introduction of bias by the researchers or by any participants who are collecting data in the study. And whether these tick reductions actually affect cases of Lyme disease in our communities is almost never studied. So we have stopped at the point of saying, well, we can reduce tick numbers that should be helpful to us, but whether it actually is helpful in protecting us from tick-borne disease is almost never studied. So that when we started the Tick Project, we had this burning question that had not been answered, which is, does killing ticks protect our health, protect us from exposure to tick-borne diseases? We were influenced by one study that was quite large. It was very well replicated. It occurred in 3 states in the Northeast, Massachusetts, New York, and Maryland. And they did show a strong experimental design and proper controls. Unfortunately, in that study, which was led by our collaborator on the Tick Project, co-author on our paper, Allison Hinckley, showed no effect of reduced tick abundance on the incidence of tick-borne disease. Tick-borne disease was just as prevalent in properties that had been treated with placebo controls as it was with properties that had been treated with the chemical agent that they used, which is called bifenthrin. So why? It's a burning question, another one. Why did this reduction in tick numbers in a large-scale, well-replicated study fail to protect people against exposure to ticks and tick-borne diseases? Well, Hinckley et al. In that study used one tick control method. As I mentioned, they sprayed bifenthrin, a chemical pesticide, in people's yards. Their study extended only either 1 or 2 years. So some properties were sprayed only 1 year, others were sprayed 2 consecutive years. And they treated individual properties. And those properties were very widely scattered about in large areas in those 3 states that I mentioned. So to try to take this to the next level, in the Tick Project, Felicia and I chose to use 2 tick control methods. I'll describe those in a moment. Our study was 5 years long. Actually, it took 6 years because of delays from the COVID pandemic, but we deployed these tick control methods in 5 years and took data actually in 6. They treated individual properties, we treated neighborhoods, and more about that in a moment. So our goal was to develop and test as rigorously as we could the efficacy of a safe and affordable means of preventing tick-borne disease at the scale of whole neighborhoods. The 2 interventions that we chose to use to control ticks had already been shown in these preliminary trials that I had talked about already to be effective in reducing tick abundance. They were commercially available. We wanted any results to be able to be used in the real world if it proved effective. So we wanted any products that we were using to be available for use following the study. And we needed them to be known, demonstrated safe for people, pets, and the environment. So the first of our 2 interventions is a microscopic fungus called Metarhizium bruneum. And I'm showing the slide of a forest floor in order to describe to you that this fungus is probably occurring somewhere in that slide, but we can't see it. It is a native fungus. It naturally occurs on the forest floors of Eastern North America. But it's microscopic, and it makes its living by attacking certain species of arthropods, and it consumes them. And this is what it looks like when a tick on the right an engorged tick. And on the left, this is the egg of a tick, picture from my lab, is covered with this gray fuzzy stuff. That's the mycelium of Metarhizium bruneum. It is eating the tick from the outside and killing it. And so this is a naturally occurring microscopic fungus that has been commercially developed. A particular strain called the F52 strain has been developed into a commercial product called Met52. So this is a fungal spores that are mixed with water into a solution that can then be sprayed with a high pressure truck mounted sprayer into the environment, as you would with a chemical spray, although this is not a chemical spray. The other intervention was one of these host targeted interventions, and this is called the TCS bait box. And it's shown with this little black box with two rodents that are not to scale. That is a gigantic chipmunk. This is a plastic box, and we used a galvanized steel shroud over the box to prevent varmints and things like children and pets and wildlife varmints from getting at these things. And you can see in front of this mouse, there's a hole that allows the small mammals to gain access to the inside of these boxes. And there's a small kind of poor quality bait that is somewhat attractive, draws the animals in, but in order to get to the interior of the box all the way through to the bait, the small mammal brushes against a wick that applies onto them a tiny drop of a chemical tick-killing agent or a acaricide that's called fipronil, which is the active ingredient in Frontline, which has been used on probably hundreds of millions of pets with a high safety record. And we would deploy a handful of these bait boxes on individual properties within our treated areas, an average of about 5 or 6 bait boxes per backyard. So where did we deploy these two tick-killing interventions? Our study was conducted in Dutchess County here in southeastern New York, where we acquired from our collaborators at the Dutchess County Department of Behavioral and Community Health maps of cumulative cases of tick-borne disease, Lyme disease in particular, from recent years, and we were able to create a map. Bob Wills helped us create this map of the distribution of Lyme disease incidence in Dutchess County. It's sort of a heat map where redder, warmer colors indicate more incidence of tick-borne disease. And we selected neighborhoods that were basically in the warm to hot zone. So we wanted to deploy these interventions in the places where the problem is the greatest. And we selected 24 neighborhoods that were all in Dutchess County and all in those warm to hot zones that I just showed. And we spent the year of 2016 attempting to recruit as many residents within each of those neighborhoods as we possibly could. We were as thorough and persistent as we could ethically be in trying to entice these people to engage with us as participants in this study. We used snail mail, we used email, telephone, we went door to door. We tried until we felt as though we shouldn't pester people anymore. And we had about 34% of the properties in a neighborhood participating. So the range was a low of 24% to a high of about 44% of the properties. And there were roughly about 100 properties within each of these 24 neighborhoods. So our study design was to divide these 24 neighborhoods into 4 different categories of treatment, where there were 6 neighborhoods in each category. So looking first at the upper left, so we would have 6 neighborhoods that had active bait boxes. So these were bait boxes that actually contained that fipronil, that tick-killing chemical on the inside, and were designed to be active, but they had placebo Met52 spray. So although the same trucks and the same truck operators and the same equipment was used, we were not spraying MET52 on those neighborhoods. We were spraying water that was actually provided for us by the Dutchess County Water and Wastewater Management Agency. For 6 of our neighborhoods, we had placebo bait boxes. So these were identical to our active bait boxes on the outside, but on the inside, they were also identical with one key exception, which is they had no fipronil. There was nothing to kill ticks on the mice and chipmunks and shrews that entered those boxes, but they had active MET52 spray. So there were 6 of those neighborhoods. Then we had 6 neighborhoods that had the active version of both bait boxes and MET52. And then we had 6 neighborhoods that were placebo controls for both of those tick killing interventions. They had just bait boxes with no fipronil and only water being sprayed. Both the residents of each of these neighborhoods and the researchers that were going out to collect data were unaware of which treatments were being used in which neighborhood they were investigating or living in. Okay, so we deploy this design of 2 tick-killing interventions with a placebo control. What are we asking? What are our response variables? What are we collecting data on? So we pestered all the participants in our study every 2 weeks throughout most of the year, with a little bit of a holiday break around Christmas time, for 5 consecutive years. So we were deeply engaged with these people for a very long time. We asked them by text or email or telephone, whatever they preferred, every 2 weeks, whether there had been any cases of tick-borne disease diagnosed in their household, including their family members and their pets, or any encounters with ticks in the preceding 2 weeks. If they said no, we left them alone, but not for very long, because we went back again 2 weeks later. If they said yes, we then followed up by requesting their permission. They were free to deny permission. We requested their permission to contact their healthcare providers to see if we could get confirmation of that self-reported case of a tick-borne disease. And then finally, we sent teams of researchers onto some of the properties within each of those neighborhoods to determine how many ticks there were crawling around in the environment and how many were occurring on the small mammals. And we did this by live trapping. We wanted to know how many ticks there were on the properties within each of these 24 neighborhoods as a function of the treatment type that they received. So here are a couple of our researchers. I'm not sure if you can see that this particular researcher is holding a mouse, a living live mouse, squirming in her hand, and she is counting the ticks on that mouse. That was one of our important pieces of information. This was done during live trapping. And we asked the question about whether those tick-killing interventions were effective at reducing the number of ticks on the small mammals, which are very important hosts and determine a lot about the abundance of ticks in the environment and the proportion of ticks that are actually carrying the pathogens that can make us sick. So I have an annoying blockage at the bottom of my screen, so I can't quite see what's on my screen. But what we looked at were the number of ticks per mouse per small mammal host in our 4 major treatment types, and that's what I'm going to show in the next series of slides. So for neighborhoods in which neither treatment was active, so these are the placebo control neighborhoods, we saw on average a little more than 5 ticks per animal host. It's the weighted mean number of ticks per animal. When in the neighborhoods in which we had active bait boxes, the number of ticks per animal host was reduced by a little more than half. So that was a statistically significant reduction, that the bait boxes were significantly reducing the number of ticks per animal host. For neighborhoods that had MET52 spray that was active, we did not see a significant reduction in the number of ticks on animal hosts. And in neighborhoods with both treatments active, both bait boxes and MET52, we saw results very similar to those for active bait boxes alone. So the conclusion we drew is that there were fewer ticks on mice and chipmunks on the properties in which there were active bait boxes in that neighborhood. We also sent our teams of researchers into harm's way, wearing their protective white suits and bringing these rolled up corduroy drag cloths to determine how many ticks were crawling around in the environment, not the ones necessarily on the small mammal host, but the ones that could get on people if they walk through that same area. And again, we're going to look at the mean number of nymph stage ticks. This is the most important stage when it comes to transmitting tick-borne diseases to us. The mean number of nymph stage ticks per time interval that we spend dragging these claws to collect ticks on the ground. And at first, I'm going to look at this in the forest habitat that many of these properties had in various neighborhoods. So for the controls, so this is where neither of our interventions were active, they're both placebos. We saw about a little over 0.3 ticks per time interval that we spent flagging on the ground to collect ticks. With bait boxes, we saw a reduction, again, by a little more than half, a somewhat more modest reduction where there was active MET52, and where we used both interventions active, the data look almost exactly like for bait boxes alone, a reduction by a little more than half. That was a similar result in kind, but not in magnitude for what we saw on lawns. So on lawns in the control neighborhoods, we saw far fewer ticks in the environment. So again, this is where neither intervention is active. There were far fewer ticks on lawns than in the forest. This is a repeatedly made observation throughout the range of Lyme disease. And again, with bait boxes active or both bait boxes and MET52 active, we saw a reduction by about half, with MET52 not showing a significant reduction. So our data told us that there were fewer questing ticks out in the environment in the neighborhoods that were treated with active bait boxes, with a modest or even undetectable effect of MET52. When we looked at the cumulative number of cases of tick-borne disease found in outdoor pets, over the 5 years that we were collecting data, we determined that there were a little over 4, almost 5 cumulative cases of tick-borne disease in pets in neighborhoods that had neither of the interventions active, those control neighborhoods. With bait boxes, bait box neighborhoods, MET52 neighborhoods, and those with both being active, we saw a reduction, a statistically significant reduction by almost half in the cumulative number of cases of tick-borne disease in outdoor pets. But finally, for people, we saw a similar cumulative number of cases of tick-borne disease in people as in pets, slightly more in people, a little over 5 over the years of the study. And with bait boxes only, with MET52 only, and with both being active, we saw no reduction in the cumulative self-reported cases, human cases in our populations. We saw no effect either on the healthcare provider confirmed cases nor on the rate of tick encounters with human occupants of those residences. So let me summarize what we learned and what might be next. So in neighborhoods that had active bait boxes, we observed half as many ticks in people's yards as with any of the other treatments. In neighborhoods with active MET52, there was either a very modest effect or none at all. Reducing ticks in people's yards with bait boxes, active bait boxes, did reduce the number of cases of tick-borne disease in outdoor pets. But reducing ticks in people's yards did not reduce the number of cases of tick-borne disease in people, nor did it reduce their likelihood of reporting an encounter with ticks. So where do we go from here? Previous research, as I've described at the beginning, that used less intensive treatments, only one of them perhaps, or only in isolated backyards rather than neighborhoods, found a similar result, that reducing ticks in yards doesn't reduce incidence of tick-borne disease in people. Our expansion to neighborhoods, which we expected might reveal a different result, did not. It did not affect human cases either. Why? Well, we don't know the answer to that. We have some hypotheses, and one of them is that perhaps if we had very high participation, rather than 30 or 40%, we had 80 or 90% of the households in a neighborhood participating, we might have seen stronger effects. We simply don't think that is feasible, that is ever likely to happen in the real world because of our efforts to recruit people in communities that know of us. They know the Carey Institute, they know Bard College, they know our research program. I think there's a high level of trust in what we do, and our 30 to 40% participation was, I think, the best that can be expected. It's possible that people encounter the ticks that make them sick when they're away from their yards or away from their neighborhoods, so that reducing tick abundance in their neighborhoods did not necessarily reduce their probability of getting sick because they were getting exposed elsewhere. In contrast, maybe the treatments were effective for pets because pets are more likely to spend time in the yards and encounter ticks there, or perhaps they spend more time in the forested part of the yards, which is much riskier than the non-forested parts of people's yards, whereas people perhaps know better and avoid the riskiest places. So if yard treatments, as revealed by the Tick Project, at the level of neighborhoods or at the level of individual properties, are unlikely to reduce incidence, what's left? Where do we go from here? It is encouraging that personal protection still works. It is effective. So the use of repellents on our skin, the use of tick-killing agents like permethrin sprays on our clothing is known effective in protecting us, as are tick checks when we come in from the out of doors, especially in the risky time of year, which is starting right now. Although there are research efforts on vaccines for Lyme disease specifically, those wouldn't protect against other tick-borne diseases, and we think it's time to invest more heavily in vaccines against tick bites. This is in earlier phases of research, but we think it's very promising that vaccines can prepare our immune systems to actually attack biting ticks before they're able to transmit any pathogens, so that we effectively kill or weaken the ticks themselves. And we think that's a promising area for future research. We also think, finally, that education is critically important so that we can facilitate early detection of tick-borne diseases and treatment of those people who fall ill at an early stage in their illness, which is far more effective than starting treatment later on. So I want to quickly acknowledge the Army, the more than a village, practically a city that it took to undertake this, this large project. We had amazing crews of field researchers who put themselves in harm's way, facing hordes of ticks on a regular basis. And as you can see from this slide, even though this is widely known to be the worst job in the world, they were extremely cheerful, with one possible exception. And every year, they were mostly new people, and every year they went and did the task and collected our data. And I think some of them may be on this call, so just please accept my deep gratitude and that of Felicia for all that hard work over a number of different years. We had a number of people that I won't name, I won't read their names, but there are their names, who were intimately involved with all phases of this research, mostly from the very beginning. We are deeply grateful to our 2,384 human participants who stuck with us throughout this whole process and put up with us bugging them every 2 weeks. We're also grateful, and we hope those human participants will forward our gratitude to their 849 pets. The bulk of funding for the Tick project came from the generosity of the Stephen and Alexandra Cohen Foundation. We had in-kind contributions by a number of different agencies at the level of the federal government, the state, the county. There were a number of private foundations and generous individuals who were gracious and generous in donating to allow us to bring the Tick project to fruition. Felicia and I are both deeply grateful to our institutions that employ us. We're particularly grateful to Josh Ginsberg, Catherine Forbes, and the entire Carey Institute, who went through all kinds of contortions to help us do this project. And Bard College behaved very similarly. And lastly, there are a few people who were engaged in other ways beyond any expectations, and we'd like to express our gratitude to them as well. So I will stop sharing my screen and somewhat belatedly start the question and answer phase for a little more than the next 15 minutes.
Great, thank you for that wonderful talk, Rick. There are lots of questions, but I encourage you, if you have questions, to go ahead and put them in the Q&A down at the bottom, or in some cases at the top of your screen, and we'll get through as many of these as we can. So Rick, we're going to maybe try to spin through these pretty fast because there are a lot. So one question about methods was for the tick species on the tick count with chipmunks and mice, were the tick species differentiated? For example, were soft ticks included?
We did differentiate in the field as best we could. We know from many years of monitoring small mammal populations at the Carey Institute and identifying tens of thousands, maybe hundreds of thousands of ticks on them, that the vast majority of the ticks that we were seeing were larval and nymphal stage black-legged ticks, Ixodes scapularis. We did not see soft ticks on these animals, and any obviously different hard ticks, other species of ticks, would have been identified in the process of doing these inspections.
Thank you. One person, and I'm not sure where this person is writing in from, says that in this person's area, the prevalence of ticks in fields and tall grass is the greatest. Did you test in a field environment, like an old field environment?
There was little representation by old field environments in these residential neighborhoods. So there was lawn, there were gardens that included shrubs, and there was forest. And we did sample in all those. We have done a fair bit of research at the Carey Institute in old fields, and our data and that from a number of other published studies show that ticks in old fields, these are grassy, herbaceous old fields, are generally quite scarce compared to forests, with shrubby old fields being intermediate. So that's what the data tell us, both locally and regionally.
There are several questions about the safety of the methods we used for various things like pollinators, for the use of the fungal spray, for example, and also what about, could there be an impact of the fipronil treatment of the rodents on predators, for example, owls and carnivores? Do you want to address that one?
Sure. Yeah, so in research that that Ilya Fischhoff, who was a postdoc on the project, led with Felicia and I and other collaborators joining him, we specifically asked the question of MET52 experimentally at field sites at Cary, whether non-target arthropods, mostly insects, but other arthropods as well, things like mites and thrips, were affected by MET52 spray. And we were gratified to see that they were not, that although ticks were reduced quite dramatically, that these non-target arthropods, generally speaking, were not affected. And in the few cases when they were, their populations bounced back quickly. So the non-target effects of MET52, and we published a paper on this, were demonstrated to be quite modest. With fipronil on the small rodents, this has been studied to some degree. The amount of fipronil that is placed on the mice and chipmunks is so small that a predator that consumed hundreds of mice and chipmunks would be exposed to a lower dose than a cat or a dog being treated by its owner. So there's very low vertebrate toxicity with fipronil, and the amounts that predators in the natural environment could have accumulated by preying on the mice and chipmunks that got treated because they entered active bait boxes, we perceived as likely to be very low and not problematic.
Thank you. There's a question about the retention rate of participants across the entire time of the study. I'll field that one if you don't mind.
Yeah, please do.
This was something we were very concerned about at the beginning. We had a lot of people telling us that we were asking too much of our participants for too long and that we would lose participants at a really rapid rate. We actually had, I don't remember the exact number, but it's in the paper, but the retention rate was in the mid to upper 90% range. We actually had remarkable retention across the study. And actually most of the people who left the study moved out of the area, and often then we were able to recruit someone to rejoin from that particular property. We just kept the property in the study. So we were overwhelmed by the enthusiasm of our participants and their willingness to put up with us and our white suits and our drag cloths and all of that. So thanks, shout out to them if any of them are on here. We gave a webinar for our participants back in March when we first had the paper accepted, and so that we've spoken to them directly. But if you're out there, you have our gratitude yet again. There's a question about MET52. We did not find MET52 to have particularly high efficacy. It didn't do much for anything, any of the response variables. And there's a question raised somewhat stridently that that MET52 has been very effective in other studies, like 95 to 99% effective in other studies, and that if studies that haven't found it as effective have typically sprayed the MET52 when it's wet out, or used it in, and not used pressure sprays, or used it in a dilute way. So could you address how we applied the MET52 and it's likely any potential reasons for the difference between our finding it's basically ineffective in most situations and other people's finding it more effective?
Yeah, it's an important question, and we were surprised to find such low efficacy. The previous studies had shown it to be effective. I don't recall ones of tick reduction, tick control by over 90%, but that may be the case. We used high pressure truck mounted sprayers, and so that is more effective than backpack low pressure sprayers in prior work. We used it according to label instructions, and we avoided spraying whenever there was precipitation either going on or expected. We waited, I think, 24 or 48 hours after any precipitation events. So it was used in accordance with how one is supposed to use MET52, and it was not very effective. It's known to break down, to deactivate under sunlight. And so all our lawn areas that were being sprayed were subject to sunlight because they were not very well shaded. But we also sprayed well into the forests that were present on people's properties where we expected that it would be more effective, but our data told us that it wasn't. So we don't know why it was less effective than in some prior studies. I should also mention that some of the prior studies didn't use a blinded placebo control. So it's possible that there was implicit bias by the researchers introduced into the collection and interpretation of data. So that is a possible contributor to this, but all in all, it was a surprising result, but it is the result we got.
Thank you. Were the number of bait boxes based on the size of the property or the size of the landscaped yard area?
They were based on the size of the property. And again, we conformed to the instructions by the producer of the bait boxes. And so, and that is how the 5.5, I think, on average bait boxes per property was derived by complying with the instructions that were based on pilot studies that demonstrated their optimal efficacy in other similar types of environments.
Right. Another question, sort of methodological one, so I'm going to get these mostly out of the way here. Were different neighborhood densities measured, and if so, was there any difference in neighborhood densities? I can take that one as well. We actually had a fairly standardized neighborhood density in order to, for us, a neighborhood consisted of about 100 properties in a certain geographic area, and so that meant that lot sizes were pretty uniform. So all the lot sizes, they were pretty typical village or suburban modest sized lots, and we had about 100 properties that defined a neighborhood, and then we recruited as many houses, as many specific properties within there as we could, and as Rick said, that was about 34%. So the neighborhood densities were very similar across, because that's where we were seeing high Lyme disease risk in that risk map that Rick showed to you, and also we set bounds around our study to make it feasible. That meant that we were working in fairly high density neighborhoods for Dutchess County. Do you want to add anything to that, Rick?
Nope, that explains it beautifully.
Great. There's a question of, are there any natural tick predators?
Yes, there are a few, and we know very little about how effective they are in controlling tick populations as well. There are some ants that eat ticks. There are wolf spiders that eat ticks, and this postdoc that looks like Lori Quillen posted the paper that Ilya Fischhoff led on non-target effects. He also led a paper looking at the effect of wolf spiders on tick abundance in controlled experimental apparatus. There's a species of parasitic wasp, a little tiny wasp that lays its eggs on ticks, and those eggs hatch into larvae that consume the tick and kill it. So there are a few things that eat ticks in nature, but this is a very poorly studied area, because although we can list a handful of species that do so, we don't have very good comparative or experimental data that would indicate whether they would be promising as tick control, whether we can manipulate populations of tick predators to encourage a stronger reduction in tick populations. It is possible that some of the variation in the abundance of ticks from one place to the next is a consequence of variation in the abundance of tick predators in the vicinity. And so the MET52, the fungi, and there are several fungi that are known to be able to kill ticks as well. Those would be considered more pathogens of ticks, but they would have the same kind of impact potentially as the predators would. So yes, it's a fascinating area, and it's one that really is asking for more study, both from a basic and applied point of view.
There's another question. These are sort of moving into a little bit of what comes next. One of them was pathogen prevalence similar across neighborhoods and treatment groups?
We don't know yet. So we collected an enormous number of ticks, and they have been being processed for the past couple of years in the molecular lab at the Carey Institute. We are nearing the finish line with having those ticks processed through PCR, polymerase chain reaction, to determine which of those ticks were infected with which pathogens. We don't have all those data in hand yet, but we hope to in the coming months.
Great. Let's see. There's some concern being raised about the stability of MET52. So could you just comment on that? Or I guess I could just say that there is some concern about the stability of MET52 as a product, that we had a little bit of trouble getting the commercial product that passed quality assurance and control, and the company had a little trouble with that. Other researchers have reported that it's hard to keep the spores viable. So this is a bit of an issue, and it might have been part of why we were at least as careful as any other commercial applier would be in this. We've made a lot of accommodations to make sure that everything was working at really optimum levels. And so I think what we would say is, if it didn't work in this trial, at least this version of the product isn't going to work in more standard formats, because we could go on and on about the ways that we made sure that it was applied under optimum conditions. So I think it's a real result. There's a question about what's next for us. What might be the next project? And so if it's not for us, what would you say? What are the big next steps? What would you focus on next?
For us, or in general? Also,
in general, but I mean, I don't mean to put you or me on the spot here. So what would you say are big next steps about prevention?
Well, one of the important areas that I think is suitable as a next step is to learn more about where people are when they encounter that tick that ultimately makes them sick. There was a sort of a conventional wisdom that arose early in the Lyme disease epidemic, that Lyme disease is a peri-domestic. In other words, you get exposed around your home, on your property. There's some evidence that exposure is within your neighborhood, but not necessarily on your property. But we really don't know how often we encounter that tick that made us sick somewhere else, somewhere outside of our property, somewhere outside of our neighborhood, and what we were doing. And that's difficult to get information on, because when people find a tick on them, they assume that that tick must have just found them, that it has not been on them for very long. And that assumption is actually not necessarily correct. When ticks get on you, they will often crawl around for hours, even a day, and they're figuring out where they want to sink their mouth parts into you, and they take their time doing it. So you may find that tick and think that it was something you were doing in the previous 15 minutes or half an hour, but that tick got on you 8 hours earlier. And so that makes it very hard to determine where you were and what you were doing. So that is an area in which we need much more information, because it would then be useful if we find a pattern that's different from the conventional wisdom, that we could attempt to treat ticks, control tick populations, targeted in those areas, if there are such areas that are reliably highly risky. So that's one area. And so I would say that that's, in terms of tick control and tick-borne disease prevention, that's a critical area to move into next. So I guess I'll stop there.
That's great. So there is a fair bit of chatter about possums and turkeys and some chickens as predators. Do you want to just briefly address those, and then maybe we can sort of wind this up?
Sure. So in a study led by Felicia a number of years ago, we determined experimentally in a combined field and laboratory study that opossums kill the large majority of the ticks that are on their bodies and trying to feed on them. So the possums are turning the tables on the tick. Instead of getting bitten by the tick, they bite and destroy ticks at a very high rate. So whether that means that we can somehow control ticks by manipulating opossums is a different question entirely, and one that I am skeptical of. I don't think we can move opossums around or try to increase their numbers by, say, feeding them. There are groups that have attempted to provide nest boxes for opossums that were based on a design of nest boxes that they found on the web. And it turns out that those nest boxes really work well for possums without the O in front of it, which occur in Australia. It's an entirely different organism. But for opossums, which are sometimes called possums, they don't use those kinds of nest boxes. So turkeys we've studied experimentally in the field and found with captive wild turkeys that they did not consume ticks when they were given the opportunity to do so. We don't know whether they really do very much in nature, but there is no evidence that they do. With chickens, there's also a buzz about guinea fowl. The evidence that chickens and guinea fowl actively consume ticks to the degree where they're capable of suppressing their numbers doesn't really exist. They may occasionally consume ticks, but there's no support for the notion that that keeping them is going to solve your tick problem. So again, not that well studied, but the guinea fowl, chickens, turkeys, other fowl are not going to be a panacea for controlling ticks on our properties.
Great, thank you. If you're willing to stick this out for a couple more minutes, there is a question, a couple of questions have come up about the MIT study using CRISPR technology, mice against ticks and that kind of thing. Could you just kind of quickly highlight what that is for people and then tell us what you think about that?
Yeah, if I can be quick, it's complicated. So there's a study being proposed in which there have been pilot studies undertaken where a group from MIT is interested in releasing genetically modified white-footed mice, starting on largely uninhabited or completely uninhabited small islands off Cape Cod near Nantucket, with a goal of eventually releasing them on Nantucket at a later stage, where these mice are genetically modified such that their immune systems prevent them from getting infected with the Lyme disease spirochete, Borrelia burgdorferi. So they're normally normal mice that are not genetically modified are extraordinary places for Borrelia burgdorferi to proliferate. And that's where many of the ticks that get infected acquire their infection. The ones that are dangerous and can make us sick get their infection largely from mice or chipmunks or shrews. So if we could turn the mice from being what we call a reservoir of infection into becoming a non-reservoir of infection, so that any tick biting these mice do not pick up the pathogen, then we could potentially protect human health. There is technology that allows genes to be spliced into these animals that would, in the lab, I understand that they can create these immune mice that are not susceptible to infection and can't infect biting ticks. I'm concerned about potentially unexpected, unintended effects of genetically modifying mice on their behavior, potentially their susceptibility to infection by other pathogens that they may share with us. We did a study, Felicia and some colleagues from the University of Pennsylvania, where we used a vaccine to immunize mice against Lyme disease, and we detected some interesting behavioral differences in those mice where they foraged in a different way. They were less risk averse than the wild type mice. So there could be, and those unintended, unexpected effects might not matter, but they might matter a lot. And we have very little ability to predict that ahead of time. I'm also a little bit concerned that in any other scenario, for instance, on the mainland or on a large island, the potential for wild type, so not genetically modified mice, to move in and swamp the modified population is enormous. And the mice move around rampantly all the time. So it could be that if it is found to be effective and safe, that it might only be usable in a very specific, almost a boutique kind of environmental setting, which would be very small islands where there is no immigration by mice from elsewhere. So I think there are concerns with it. I'm intrigued by the technology, but I think we need to proceed extremely carefully. And they are doing things responsibly, to my mind, in the sense that they're engaging the communities that would have these genetically modified mice running around at an early stage, very thoroughly and repeatedly. So, and they're willing to pull the plug, or so they say. If there's resistance, then they aren't going to try to impose their will on these communities, and I think that's a good thing.
Thanks for that. I know that's a very difficult thing to explain, and as usual, you did a wonderful job with that. I'm afraid we're going to have to cut off questions here because just to be sensitive to people's time and Rick's as well. I will say that the products that we use, the Met52 and the tick control system bait boxes, are both commercially available. There were some questions about that. Met52 is not. Has had some spotty commercial history where it's sometimes available and then sometimes very hard to find. The tick control system should be easier to find if you're interested in that. And there's information about both products on the Tick Project website, which Lori has helpfully provided in the chat, so you can grab those before we end up. I would like to close by acknowledging our dismay that we weren't able to get to all of those questions. So I'm just hereby going to volunteer Rick to do some other webinar in the future just to answer your Lyme disease questions. I'll moderate and you can have at it because there were a lot and I really apologize we couldn't get to all of them. I also want to thank, again, all of the many people involved in this study, our funders, our partners in all of this, and most importantly, our participants who put up with us for all those years. We really could not have done this without them, and we owe them a great debt, all of us, for what we learned from the study. And then I'd finally like to close by thanking Leslie Tumblety and Lori Quillen for helping us set up the tech tonight. It always works flawlessly with them involved, and we're very grateful. Rick, did you want to add anything to close?
No, I echo all those acknowledgements. It's been an incredible group of supporters and colleagues.
Great. All right. Thank you, everyone. There will be a recording of this made available to you from the Carey Institute. It'll be posted online for about a month. And there is a results page on the Tick Project website. You can also access the paper there. And again, Lori Quillen has been posting these papers in the chat for this whole time, so you have access to more than you could possibly want to read about Lyme disease and tick control. So grab what you can there because that will disappear when the webinar is over. So I'm stalling now a little bit if you're wanting to click on these things. And we will sign off, but leave this up for just a moment before you, so you can grab those links. Good night, everyone.
Thank you very much. Thank you, everyone.